So let's kind of imagine, I mean, we've been seeing this already. You have the cell body of a neuron, and then at one end, you're going to have the receptor, right? This is basically the receptor, the dendrites of the receptor. And again, I know it kind of has these glyphs, or these artifacts that happens when I draw, but hopefully everybody kind of understands what I'm getting at here. And then at the other end, you're going to have the axon and then the axon terminals, right? And we said that this might be enclosed inside of a ganglion, for example, if we're talking about sensory neurons. So this end, the end that's going to pick up on the stimulus, that is the receptor that we're talking about. So these are essentially still neurons. Okay, that's what we're referring to here with these receptors. These are neurons. Okay, and we're going to have, we can classify these specialized neurons into different classifications based on morphology, based on different things, and that's what we're getting at right now. So what we're saying here is that the sensory receptor part of the neuron is going to be located on the peripheral end of a sensory neuron. Okay, and we're going to deal with 3 different morphologies when it comes to these sensory receptors. So again, we're talking about the peripheral end, just the receptor part, okay, that's going to pick up on that information. So 3 different types that we're dealing with. We have simple receptors. Okay, so simple receptors are going to be free nerve endings. These can either be myelinated or unmyelinated. Okay, now if I'm saying that these are free nerve endings, going back to the last semester when we were talking a little about the skin, what would be an example of a simple receptor, therefore, where we have free nerve endings? And we're talking about the skin. Does anybody remember a specific name or a specific type? Free nerve endings is the key. So if you all remember the picture of the dermis that we had, and then it was showing you the different layers, the epidermis, the dermis, and then on one of them it showed you a type of sensory receptor that was located in the skin. So I'll give it to you all. The nociceptors. Do you all remember the nociceptors when we were talking about the skin? So the nociceptors are receptors that can detect what? Do you all remember? Pain. Pain. So nociceptors. Okay, so if you don't know that, that's fine. That's why we're doing this now, right? If you don't know this information, you're okay, but start studying. This is really important information. So yes, simple receptors are like those that we find on the skin, and the nociceptor is a great example of that. Okay. What's another type of receptor? Well, we have complex receptors. Okay, what's so special or what's different about these complex receptors? Well, these are a little bit different than the simple ones in that the nerve endings are not freely, just out there. They're actually going to be enclosed or encased in a specialized connective tissue. So do you all remember when we were talking about the skin and there was this type of neuron that looked like this, and then it twisted like into this weird twisty thing like that? That was the Pacinian corpuscle, if you remember. Okay, that would be an example of a complex receptor. Okay, because its nerve endings are not going to be free. It doesn't have free nerve endings at its receptor end. Okay, its receptors are going to be encased within specialized connective tissue. And then the last type of receptor that we can classify here is called a special sense receptor. Okay, and special sense receptors, as you might haven't already thought of it, these are the ones that are involved with the special senses. And that's what we're gearing up to on Thursday to talk about when we talk about vision and hearing, right? The special senses. So again, the special senses are going to work, they function because we have neurons, and these neurons, these specialized cells, can be classified as having these 3 basic types of receptors. We have simple receptors that some of these specialized cells are going to have. We have complex receptors, and we have special sense receptors. And we said that these specialized nerve cells or receptor cells are going to release neurotransmitters to send their messages in order to propagate some type of signal. Okay? And an example that we can give for a special sense receptor would be, for example, the rods and cones of the eye. Okay? Those are specialized cells. Okay? And then also the hair cells of the ear. That would be another type of specialized sense receptor cell. Okay, so, just taking a step back again and talking about these nerves and these receptors in general. And I guess sometimes it's easiest to conceptualize this when we talk about pain receptors and pressure receptors on the skin, because they're the most, like, I guess, logical to think about versus sight, which is a little bit more abstract. But when we talk about these receptors, we need to also discuss something called the receptive field. Okay, as I discuss this, hopefully you're going to start thinking about the lab that we did the other week. Okay, so again, the area that's going to be monitored by a sensory receptor is going to be called the receptive field. Okay, and we're looking at an example here of the skin that's being monitored by two different neurons. Okay, and each one of these neurons with their receptors at the very end are going to monitor, because again, these endings only reach out this far all the way out to here, right? So they physically only have the ability to monitor this specific area of the skin. Okay, so this would be, for example, receptive field number 1 in this image. And then we have another receptive field number 2, which is being monitored by this ending here. Okay, so this is just an example of what we might have on the skin. Okay, and again, the size and the density of a receptive field will then dictate how sensitive you are to a particular stimulus in that region of the body. Okay, so now, once again, the example that I'm going to give you all is the lab that we did last week when you were testing out the 2-point touch. You all did that, right? The 2-point touch thing? Yeah. Okay, cool. So with the 2-point touch, you basically had that tool that you touched upon your fingertips or different areas of your body, and you were able to change the gap that was between the two pointed ends, and you place it on different areas of your body. Now, if you placed it in an area, for example, on your back, where the receptive field was very large, and you came in with a 2-point touch, right? And it was this far apart, and the area that was being monitored was like, let's actually do this. Let's imagine this whole area is being monitored by just one receptive field. You know, so even if you came in here with a caliper that was that far apart, you would still just feel it as one point. Okay. But, you know, if the area is being monitored by a smaller and more numerous receptive fields, bringing in a caliper that's only that far apart, for example, you'd be able to feel it as two separate points. Okay, and again, the example that we give is that your fingertips are going to have a high density of independent receptive fields. And therefore, your ability to discriminate between fine detail at the fingertips is so much more when we compare that to an area like your back. So I think you all tried like the fingertips, the anterior portion of your forearm, the back of your neck, different areas like that. And hopefully you were able to view this concept in practice in the lab. Okay. All right, so what about the different types of receptors? Okay, so we have several different types of receptors. This is a little table that shows the different classifications of receptors that we have. And again, we were just discussing the classification of receptors based on their peripheral end, right? Whether they're enclosed, whether they're not enclosed. And then we talked about if they're a special sense receptor. So now we're talking about the functional classification of general sensory receptors based on what they're sensitive to. Okay, so this is what they're sensitive to. Okay, so each type of sensory receptor is going to be sensitive to only one specific type of energy or only one specific type of stimulus. Okay, and we can divide the different types of sensory receptors more or less based on the table that you see up here in front of you. So the first type that we'll talk about are the nociceptors. So this would fall under the classification that we were just talking about earlier as a simple receptor because their nerve endings are free within the skin, within the integument. Okay, but it would also fall as a functional class of receptor as a pain receptor, right? Because these specifically pick up on pain, these nociceptors. These nociceptors, they don't pick up on chemicals. they don't pick up on pressure. They don't pick up on other things. They only pick up on a specific type of stimulus.
So you said
Pain.
there's simple and what else?
It would fall under simple and based on the functional class, pain or nociceptor, which is the fancy way of saying it. If we were to talk about the receptors that are found on the skin that detect pain, right? So nociceptors. Okay, so again, these receptors are going to respond to damage. They only respond to damage or damaging stimuli that's interpreted by the brain as pain. Okay, now these are a somewhat unusual class of receptors because we have different types or different subtypes that are going to respond to different modes of damage or different modes of stimuli. Okay, so don't let that confuse you. What I'm talking about here is that we have nociceptors that are specialized to detect, for example, damage that's caused by temperature. Okay, so that's why you would feel that hot burning pain if you were to get scalded. Or at the same time, you might feel the pain of something that's really cold, right? So a very specific type of nociceptor are those that would respond to the damage of temperature pain, okay? But then there would be also nociceptors that would be able to pick up on mechanical damage, so like crushing pain, okay? And then there's other nociceptors that would be able to pick up on chemical damage as well. But we're still talking about damage, okay? Don't let that confuse you with thermoreceptors and chemoreceptors and mechanoreceptors. These don't pick up on actual damage. They only pick up on the sense itself, the sense of temperature, the sense of chemicals, the sense of pressure, but not damage. The nociceptors are a specific type that are able to detect damage that comes from any one of these other classifications. Hopefully that makes sense. Now, these are also interesting in that they're typically only active or awake or stimulated when there's actual tissue damage. Okay, and then we have two broad types of nociceptors based on speed. Okay, how fast they propagate their message. And what dictates the speed of a signal when we're talking about the nervous system? Does anybody know what dictates the speed? Myelination. Okay, so that's how we're going to differentiate these two types. So type A fibers of the nociceptors are those that are considered myelinated, and they're going to send fast pain. What is the type of fast pain that we would say? For example, pinching pain, or a cut pain, or a needle prick pain, or something like this. It carries the sensation of fast types of pain. Okay, the type C nociceptors, these would be unmyelinated. Okay, and they're going to carry the types of sensation of slow pain. So kind of imagine, if you will, yeah, well, I'm not going to even go there. So yeah, type A fibers, we said myelinated, fast pain, type C fibers, unmyelinated, slow pain. So type C fibers, a couple of examples that I can give are like aching pain, okay? Like a bump or like a bruise, something like that. The type of fibers would be type C, okay? So those are the nociceptors. And again, where would we find those mostly? On the skin would be a good place, right? Okay, maybe some certain organs and stuff like that as well. Okay, what else do we have here? We have thermoreceptors. Okay, now these thermoreceptors, they're going to respond to temperature. These are usually also going to be free nerve endings. So these are also usually considered simple. And these we can find in places like the skin, in the dermis, in the liver, in the hypothalamus. Okay, we'll find thermoreceptors in those areas. Okay, chemoreceptors, that's another type of receptor. Chemoreceptors are going to respond to chemicals. So they respond to water or lipid-soluble substances. When we talk about these chemoreceptors, we're not really talking about outside the body. We're not talking about being able to detect chemicals on your skin per se. We're really talking about them being able to detect chemicals within your body. So like, for example, within some of your arteries, you're going to have chemoreceptors that are able to detect the levels of oxygen and carbon dioxide. Okay, so those types of receptors that are able to do that are chemoreceptors. Now, later on in this semester, we're going to talk about the urinary system. In the urinary system, in the juxtaglomerular complex, we're also going to have a type of chemoreceptor that's going to be able to detect different chemical concentrations of urine, okay, in order to either concentrate or dilute urine. And then the last type of or not the last type, but almost the last type of receptor here, are mechanoreceptors. Okay, now mechanoreceptors are going to respond to mechanical stimuli. And the way that they do this is by responding to the distortion of their cell membranes. Okay, so when their cell membranes become distorted, they're going to go through some type of biochemical reaction. Okay, and at the end of that biochemical reaction, we're going to get the opening and the closing of specialized membrane channels, which will cause depolarization, which will then propagate the signal to the nervous system and the feeling of pain. Okay? Now, of the mechanoreceptors, we have a couple of different types. We have proprioreceptors. Okay, so proprioreceptors, they're going to monitor the position of your muscles and the position of your joints. So if you put your arm up into the air, you can feel the position of your arm, the position of your joint in the air, because you have specialized proprioreceptors, okay, within those structures. Now, we talked about a specialized proprioreceptor last week, I believe, when we talked about the stretch reflex. Do you all remember within the muscle? What was the name of the receptor within the muscle? Does anyone remember that one? Muscle spindle. We talked about that, right? Muscle spindle?
I
No?
don't think so. I don't think so.
No? Well, look it up. It's an interesting topic. A muscle spindle is a type of mechanoreceptor, okay? And it's located in your muscle. It's a type of proprioceptor, and it has the ability to detect stretch in the form of positional change. Okay, so yeah, so the muscle spindle is an example. Another type is a baroreceptor. Okay, baroreceptors are going to detect the stretch in the walls of blood vessels. Okay, also in portions of the digestive tract. Okay, and then the last type that we have here, tactile receptors. Okay, now these are going to provide the sensation of touch, pressure, and vibration. Okay, now these can be extremely, extremely sensitive, in which we would call them fine touch receptors, or they can be somewhat crude, or we would call them crude receptors. Okay, now as you might have already imagined, you have many of these types of receptors, of these tactile receptors on the surface of your skin. Okay, there's also many of these types of receptors on the surface of the tongue. Okay, now the cells that are involved in hearing, okay, there's actually these cells in your ear that are called hair cells. Now they're not actually hair, but they're called hair cells. Now these hair cells that again are involved in the sense of hearing are a type of mechanoreceptor, and the subtype being a tactile receptor, which is able to pick up on vibration. Okay, and it picks up on sound in the form of vibration, and it converts that into information that we understand as sound in the brain, right? And then the last type of receptor that we're going to stick down here are photoreceptors. Okay, and photoreceptors are a type of receptor that we find in the eye, and they have the ability to respond to electromagnetic radiation, which is a fancy way of saying light, and they are found in the eye. Okay, so these are, again, different types of receptors based on their functionality. All right, so thermoreceptors, chemoreceptors, mechanoreceptors. When we talked about the mechanoreceptors, we talked about three types: the proprioceptor. Okay, we said the muscle spindle was an example. The baroreceptor. Okay, they detect changes in pressure within the walls of blood vessels usually. And then the tactile receptors, which we talked about fine touch versus crude touch. Okay, and then the photoreceptors, which we said was a specialized type of sensor or receptor that we find in the eye. If you've heard of the rod and the cone of the eye, those are the photoreceptors of the eye. And then the nociceptors, I think we already mentioned. We said those are pain receptors. There are two different types, type A, type C. The difference between the two, myelinated versus unmyelinated, faster versus slower, fast pain versus slow pain. Okay, now really quickly here, receptors can also be classified on how they respond. Okay, how they respond. So some receptors are always on. They're always active. Okay, they're always ready to pick up on stimulus. Okay, those are called tonic. Okay, and so an example of a tonic type of receptor would be like smell. Okay, so we're always ready to pick up on those types of stimuli. Okay, and then whenever they get stimulated, what they do is that they would increase or decrease their action potentials in order to send their signal. Okay, so an increase in the stimulus, you get an increased frequency, increase in action potential, decrease in stimulus, a decrease in frequency. A couple other examples, pressure and touch. And then there are phasic receptors. Okay, phasic receptors are typically not active until they become activated. Okay, so for example, pain would be a type of phasic receptor. Okay, so moving on to the special senses. Okay, so we'll do like a quick summary of the special senses, and then we'll view vision and hearing in a little bit more detail. So let's start with olfaction, okay, which is the sense of smell. Okay, and again, the special senses are going to provide information about our environment. And we said that there are going to be 5 special senses. And the first one that we're talking about is olfaction, the special sense of smell. Okay, now, this sense is going to involve a very, very close example of the structure-function relationship between your nasal cavity and the nervous system. Okay, and I want to kind of talk about how we can detect different smells and the underlying anatomy that's associated with that. Okay, so the sense of smell is associated with the olfactory organ. The olfactory organ is a paired structure, okay, a set of structures that are going to provide the sense of smell or olfaction. Now, these are located in the nasal cavity on either side of the nasal septum. They're going to cover the inferior surface of the cribriform plate, the superior portion of the perpendicular plate, and the superior nasal conchae of the ethmoid. Okay, so this is a nice cross-section of what we're talking about here. Okay. So,
Can you go back to the other one for just a second?
any questions?
No, just what to write.
Oh, I'll post it.
Okay.
Yeah, but that's fine if you need me to stay here. But just so that you don't get nervous, I'll post it. Okay. You're welcome. Yep. Okay. So here's a nice cross section here, right? Of the nose and the nasal cavity and a little bit of the brain. Okay, so a lot to kind of digest here, right? So let's kind of break it down and discuss what we're looking at here. Right? Valentine's Day is right around the corner, isn't it? Nice little rose here. So when you inhale through your nose, okay, what's going to happen here is that the odorant, the chemical that we're inhaling, which is going to be floating in the air, in this case, it's the nice smell of a rose here, is going to be carried into the nasal cavity. Okay, the odorant is carried into the nasal cavity with the air that we breathe in. Okay, now we're going to find within the nasal cavity, there are several important structures. Okay, now if you remember, we were talking about certain bones of the nasal cavity, including the ethmoid, the sphenoid bone. And these bones help to form the nasal conchae. Okay, these turbinates within the nasal cavity, as you can see here, these grooves that kind of look like shells. That's where they get their name from. So that air is going to enter the nasal cavity, and these turbinates are going to cause the air to eventually become turbulent, and it's going to direct some of that air into the superior portion of the nasal cavity that we can see here, okay, up above. Okay. And again, this is the top of the cranium or the bottom of the cranium here, and then this would be the nasal cavity. So there's obviously a bone that separates the nasal cavity from the brain, right? What's this bone called again? The ethmoid bone. Very good, the ethmoid bone. And if you remember last semester when we were talking about this ethmoid bone, we said that we're going to have these small perforations within the ethmoid bone. Do I remember what those are called? The cribriform foramina. Okay, and that plate was called the cribriform plate. Okay, so essentially what we're saying here is that as the air with those chemicals enters the superior portion of the nasal cavity, the superior portion of the nasal cavity is going to have a nasal mucosa. It's going to have a nasal epithelium that contains specialized organs with these receptor endings that are going to pick up on these different chemicals. Okay, it's going to pick up on these different chemicals. And it's going to send these signals past these nerves into this large bulb and eventually into the brain, where we're able to differentiate and pick up on the different smells that we're smelling and detecting. And again, don't forget, ethmoid bone, cribriform plate. So each one of these olfactory organs, and again, the olfactory organ is the thing that we're kind of highlighting in this purplish color. It's a paired set of organs that are located inside, right, in the nasal cavity. Okay, it's just like this nice epithelium that's going to have these specialized receptive ends. Okay, so each one of these olfactory organs, and I'm going to show you a close-up now. Okay, so this is what we were looking at just now, right? And what we're doing is we're taking a little box cross-section and we're blowing it up here, and this is what it would look like. So this area down here is the interior surface of your nasal cavity. Okay, and then you can see these little tentacle monster things. Okay, that's just the nerve endings for the receptor cells. Those are like the dendrites. And they're going to stick in the nasal epithelium, right? Into the nasal epithelium. Okay, so again, what we're talking about here is that this structure that we're talking about with these cells and everything, that's the olfactory organ. And we're saying that the olfactory organ is going to consist of a layer of olfactory epithelium, which we see here, and underneath the olfactory epithelium, we're going to find an underlying lamina propria. Okay, now the lamina propria is going to be comprised of connective tissue, also blood vessels. We might also find some nerve endings, but also pretty importantly, we're also going to find these glands that are embedded within the lamina propria. Okay, now these glands are mucus glands, and these mucus glands are going to secrete copious amounts of mucus into the nasal cavity. What that mucus does is that when those chemicals enter the nasal cavity, those chemicals are going to dissolve in the mucus, and they're able to basically bind to specialized receptors that are located at the endings here.
So the
Okay.
olfactory gland is what produces the mucosis layer?
Which one is it again? The olfactory gland?
Yeah.
Yes. Yes. That's what we're saying here. It's a mucus gland, basically. Yeah. All right. So essentially, the mucus is going to dissolve those airborne odorant molecules. Okay. So now we have one of those chemicals that's going to bind to this dendrite. Okay, to this receptor, this dendrite. Okay. Okay. Now let's zoom in. Okay, so this was the mucus, right? That was the inside of the nasal cavity. And this is like zooming in to a little piece of the dendrite of the axon. Okay, so what are we saying happens here? Okay, well, what we're basically saying is that, again, this cell, this olfactory cell, okay, this blue thing here is an olfactory cell, right? It's an olfactory receptive cell. This is the receptor portion of the olfactory receptor cell. Okay, and this is the actual olfactory cell itself. okay? And what we're saying here is that embedded within this nasal epithelium that we're talking about, we're going to find these olfactory epithelial cells, and these olfactory epithelial cells, their endings are basically going to bundle together. Okay, they bundle together. One end, we said, is going to extend into the mucus, and the other end is going to send the information to the olfactory bulb. Okay, and the olfactory bulb was this structure that we see here in yellow. Okay, so essentially this cell begins here, it travels through here, right? You can kind of see it here as it travels through the cribriform plate, and then it synapses with the cells of the olfactory bulb. Okay, and these we said are the olfactory receptive cells. And when we're talking about this in a whole, in general, this is actually cranial nerve number 1. This is the shortest cranial nerve, just from here to here is cranial nerve number 1. And cranial nerve number 1, we said, is the olfactory nerve, and what it does is it sends the signal of smell from that olfactory epithelium to the olfactory bulb. Okay, and then from the olfactory bulb, it's going to send the signal to the brain. Okay? And again, what happens here? We said that when certain odorant molecules dissolve in the mucus, they're going to bind to these tiny little receptors at the ends of those dendrites that I just showed you. So now we're really zoomed in here. We're like super, super microscopic. We're really zoomed in. And we're showing that you, remember that rose that we were smelling? Well, that rose had a little volatile chemical that was floating in the air. It dissolved in our mucus, and we had a specialized receptor protein, and we have thousands of different types of receptor proteins that will only bind to specific molecules. Okay, if you don't have the receptive protein for that smell, you're probably not going to be able to smell that chemical. Okay, so we have thousands of different unique types. So we're just saying here that this odorant molecule is going to bind to the receptive protein. Okay, and when that happens, we're going to have a chemical reaction. Now, maybe a little bit later, in a future class, when you start learning more about biochemistry, you'll learn more about these processes. But essentially, bound to this receptor protein, we're going to find another inactivated enzyme, another inactivated protein that's called an inactive G protein. So the binding of this odorant chemical onto the receptor protein essentially is going to activate this inactive G protein. Okay, now the activation of this G protein, okay, basically is going to turn this site into an active enzyme site. And that's going to cause ATP to be converted into cyclic AMP. Okay, now cyclic AMP will then bind to another channel. If you recall, we talked about sodium channels that we also find on the plasma membrane. So when cyclic AMP binds to a closed sodium channel on the surface of these olfactory cells, it's going to cause the channel to open, and therefore sodium enters the cell. We call that depolarization. The signal has now started, and that's going to send that signal like we just mentioned. You see, if sufficient depolarization occurs, we get an action potential, and that signal that we're talking about of the chemicals that we're smelling here will be propagated through cranial nerve number 1, the olfactory nerve. Okay, and it'll reach to the olfactory bulb here. Okay, question?
So is ATP what activates the G protein then, like the conversion to cyclic AMP, or what is it that activates it? The odorant?
Yes, the odorant is what's going to activate the G protein molecule. The G protein molecule is what's going to then convert the ATP into cyclic AMP. The cyclic AMP is what then activates the sodium channel to open up, which causes the depolarization. And then was there another question?
Thank you.
Okay, so that's what's going on here. And what's going to happen is that signal is going to be sent to the olfactory bulb, like we mentioned. And then one other thing to talk about in this image before I move on, you'll notice that you also find basal cells. Remember, basal cells are stem cells. And these stem cells are going to be dividing constantly in order to replace the worn out, the other worn out cells. Okay. You can see one of these stem cells is already almost developed into a mature olfactory receptor cell. And then remember that not all of our receptor cells, because receptor cells are nerve cells. And we said nerve cells don't regenerate that well. Once you damage a nerve cell, it's pretty much done for, right? One of those rare exceptions are the olfactory receptor cells, because these actually regenerate on their own about every 10 days. Okay? That's a very, very rare instance of neuronal regeneration in adult humans. Okay, so again, we said that the signals would then travel to the olfactory bulb, and then from the olfactory bulb, it would pass eventually into the brain. And then don't forget that there are perforations within the ethmoid, which we call this specific area of the ethmoid, we call the cribriform plate. The openings are called the cribriform foramina. And we said that the short axons between the nasal epithelium to the olfactory bulb is actually the shortest cranial nerve in the body, which is considered cranial nerve number 1 here. Okay, and then we said that the axons from the olfactory bulb would then extend to different regions of the brain. So you can see here on this image, they did this on purpose, where they kind of forked the nerves, right? There are different pathways. And what that's trying to tell you is that different areas of the brain would receive the same signal. So, for example, there would be an area of the brain that would be in charge of analyzing the chemical structure of what you're smelling. It would be responsible for maybe categorizing the similarities of different smells. So if you smell something like different types of trees, they all smell woody, right? Or if you smell different types of citrus fruit, they all smell citrusy. So one region of the brain categorizes different types of smells. There'd be another area of the brain that would mediate our conscious awareness of what we're perceiving the odor to be. There would be another area of the brain that would be involved with emotional and autonomic responses with the odors. There would be another area of the brain that would be responsible for motivation or memory. For example, maybe the thing that you're smelling might be like a food that you enjoy when you were a kid, and it makes you feel happy. It causes you to salivate. It makes you want to eat it. It makes you think of memories of your childhood that were associated with eating that food. So a lot of complex things are going on when we're talking about the sense of smell and the brain. And then finally, some of these odors are going to be stored in long-term memory. And then these usually have strong connections with emotions. We usually have strong emotions with the things that we smell, right? We're usually very attracted to things that we smell or very averse to other things. Okay? So that's the sense of smell. Okay, so important things here, don't forget the olfactory receptor cells are those modified neurons. We talked about the basal cells, the lamina propria, and then the olfactory gland. Another name for the olfactory glands, Bowman's glands. Don't confuse that with Bowman's capsule, which deals with the kidney. Okay, we showed you this image of the cross section. You know, what is an odorant, right? You guys can read that one. And then we went through those steps, and that was it for the sense of smell. Okay, let's move on to gustation, which is the sense of taste. All right, so gustation is going to be the special sense of taste. So this is going to provide us with important information about the foods and the liquids that we ingest. So gustation is largely going to be a function of the receptors that are located on the tongue. Okay, so when we're talking about the receptors, ingustation, we're talking about the receptors on the tongue. What do we call those receptors? Does anyone know? The special organ or structure for gustation? Taste buds, right? Taste buds. So these taste buds are what are the receptors for taste, right? For gustation. So before we get there, before we talk about the taste buds, we're going to talk about something called lingual papillae. So you probably have noticed, sticking your tongue out in the mirror or whatever. Here's a couple of different images here of the different structures that you're going to find on the surface of your tongue. Okay, this is actually a cat's tongue, but in any case, you all have seen your tongue in the mirror. So the tongue is going to be covered with epithelium, and this epithelium is going to have numerous projections that are called lingual papillae. Okay, remember, any type of projection is called a papillae. It literally means nipple-like projection. So lingual papillae are going to be these structures that we see here. Okay, again, this is another different type of a lingual papillae. Okay, another type here and another type here. It's not the same as a taste bud. We're not there yet. Okay, we're just talking about these projections that we find on the surface of the tongue. So the tongue has 4 basic types of papillae. Okay, the first type that we're going to talk about is called vallate papillae. Okay, vallum means wall. Okay, these vallate papillae are relatively large. Okay, each papillae are actually going to contain up to 100 taste buds. So as you see here, okay, and then this is the vallate papillae. Okay, so as you can see here, okay, these are the vallate papillae. We have about 10 to 12 that are located on the posterior of the tongue. Okay, these large projections are these vallate papillae. And if we were to see what these vallate papillae are comprised of, they're comprised also of these structures called taste buds. And that's where we have the sensory receptors for the sense of taste. Okay? And again, these are called vallate papillae, about 8 to 12 large papillae, shaped in about, they're shaped in a V shape in the posterior margin of the tongue. And then these each contain numerous taste buds. Okay, as you can see here, a lot of taste buds, right? Lots and lots of taste buds. Okay, the next type of papillae, foliate papillae. Okay, so the foliate papillae are going to be found mostly on the lateral margin and posterior region of the tongue. Okay, what do they look like? The foliate papillae are these here. They're located on the side. Okay. Located on the side. And then these also have taste buds. Okay. Now the next type of papillae are the fungiform. Okay, so these here, as the name suggests, are mushroom shaped. Okay, and these are going to be scattered among the filiform papillae. These also contain taste buds. So going back to this one, fungiform papillae, they also contain taste buds. And then lastly, the filiform papillae. Okay, phylum means thread. You know, the filiform papillae, these are actually the most numerous. However, these are the ones that do not contain taste buds. These are purely mechanical. And what they do is they help to provide friction in order to move the food around your mouth. Okay, now these are actually particularly pretty large in cats. Okay, and that's what gives cats, and again, going back to this one here, this would be an example of a filiform papillae in a cat. And that's what provides a cat's rough texture on their tongue. That's kind of like sandpaper, right? It's used to groom themselves because of the filiform papillae. We also have them, right? All of these is an example of the filiform papillae here. But they are not as long and exaggerative in humans. Okay, so again, we said the 4 different types: vallate papillae, located on the back, 10 to 12, most numerous taste buds. And then one other thing about the vallate papillae, no, actually, one other thing about all papillae in general, not all papillae, all taste buds, I should say, rather. So we just said vallate papillae, foliate papillae, fungiform papillae, they all contain taste buds, right? The only one that does not contain taste buds is the phyloform. Okay, we're good. Now, all three of these, the taste buds that they have are all the same. So any area of the tongue that we just talked about is able to detect sour, bitter, salty, and sweet. Any area of the tongue is able to detect those four taste sensations. Okay? So again, there's no difference, there appears to be no difference in the structures or the primary taste sensations provided by the different papillae that we talked about. And then again, the primary taste sensations, sweet, salty, sour, and bitter, are all gonna react to these same taste buds. The only extra one that we're gonna talk about, which is umami. Okay, so umami, which I guess we associate that nowadays, if you've heard, with like MSG. It's that really, really savory flavor. That's found only in the vallate papillae, those types of taste buds for umami flavors. Okay, but all the other ones, like we said, they're all able to feel sour, bitter, salty, and sweet. They're all able to detect that. Okay, now there's also in the pharynx, as you can see here, in the throat, pharynx, right? We're also going to have receptors here as well. And these receptors are water receptors. And these are going to regulate water intake and water balance and blood volume. Okay, and they're able to detect liquids. So that's one of the reasons why if you drink a really cold beverage, these would become like overactivated, and you can actually fool yourself into thinking that you've quenched your thirst with a cold beverage when you're actually still dehydrated. But in any case, those are for water receptors. Okay, so a little bit more detail here about these taste buds. So, like we just mentioned, those different papillae are going to have different taste buds and taste receptors. So embedded within each one of those fungiform, foliate, and vallate papillae, we talked about specialized epithelial cells that were called taste buds. Now, each taste bud is going to contain between 40 to 100 taste receptors. Those actual taste receptors are called gustatory cells. Okay, so this is an example of what we're talking about here. So here we have one of these papillae. If we were to look onto the lateral aspect, we can see the taste buds. Okay, this image here is showing you a histological slide of the same thing. Okay, this is one of the papillae, and then you can see the taste bud here in the center. Okay, and then here we're looking at only one of these taste buds, one of these structures. That's what we're looking at here. And then what we've done here is we're showing you an artist's representation of the same thing down here, so it's easier to read, it's easier to understand. Okay, so what we're saying here is that embedded within each one of those papillae, we're going to find a taste bud. And each taste bud is going to be comprised of 40 to 100 of these specialized gustatory cells. So this long cell that you see here, that's one of those gustatory cells. That's a specialized receptor cell that plays a key role in taste. And you can see that at the very, very end of the gustatory cell, you have these projections called taste hairs. Those are actually microvilli, okay?
500 gustatory receptors in a
You know, different books are going to say different things. The resource that I got was 40 to 100. Yeah, 40 to 100. So again, some of these papillae might only contain a few of these taste buds. Other papillae might contain hundreds of taste buds. Okay, and again, they're made up of these structures and these gustatory cells. Okay, now these gustatory cells, as you can see, are essentially elongated modified cells. Okay, these are elongated cells. And at the very, very end, we said we're usually going to find these structures that are called taste hairs. These are microvilli. And we'll see that we get a bunch of these taste hairs embedded within this pore, which we're going to call a taste pore. And you can see that down here as well, what that kind of looks like. Okay. And then again, if you're looking at this image here, at that end there, you're looking at the taste pore, right? That would be that end there. Okay. So these gustatory cells, like I mentioned, they're going to pick up on the different chemicals from the different things that we consume, and they're going to send that signal down onto the axons of sensory neurons that are located on the opposite end. Okay, now another interesting thing here about this structure is that these gustatory cells, they're only going to last about 10 days. They're constantly being replaced. Okay, and again, the reason why they're constantly being replaced is because a lot of mechanical damage occurs in the mouth. Okay, and they're being replaced constantly by stem cells or basal cells, as you can see here. Okay, now when one of these gustatory cells is stimulated by a dissolved chemical in the mouth, it essentially is going to release neurotransmitters. And these neurotransmitters are going to travel and activate the dendrites of these sensory gustatory neurons that we see on the opposite side. And these sensory gustatory neurons will then send the signal to the gustatory centers of the brain, where we then pick up on the different tastes. So each one of these gustatory cells are going to respond to 2 or more different taste stimuli. And the taste buds in all portions of the tongue, we said, are sensitive to all 4 of the 5 primary tastes. We said umami is the exception because that one is only going to be detected by the receptors in the valley papillae. Also, in general, humans are more sensitive to tastes that are unpleasant versus tastes that are pleasant. And the reason for that is because unpleasant tastes are more likely to be potentially damaging. For example, like bitter and sour is more likely to be poisonous than sweet or salty. Again, being a survival mechanism. And another thing here is that we begin to lose our taste buds as we age. And by the age of 50, 60, we've dramatically lost a lot of those taste buds, which might be one of the reasons why, anecdotally, you see sometimes older people putting a lot of salt or, stuff like that on their foods because a lot of those taste buds are gone by then. What's
Are
that?
they gone when you get older because of just mechanical damage?
Not really mechanical damage. It's more programmed through our DNA. Yep. Okay, so taste sensations, the 4 primary, please don't forget those. Umami we talked about. A couple examples like beef broth, Parmesan cheese. Water receptors concentrated in the pharynx. And then the information from those water receptors are going to be sent to the hypothalamus. Because remember, the hypothalamus is the region of the brain that's associated with thirst. Okay, so that's going to affect water balance, blood volume regulation, and also prevents the overingestion of water. Okay, we said that more sensitive to unpleasant stimuli. All right. And then the last thing here with the gustatory stuff, just to talk a little bit about the cranial nerves that are involved with gustation. This might show up in the exam as well. As you noticed, like last semester, cranial nerves and questions like that like to appear on exams and quizzes and stuff. So the cranial nerves that are involved in transmitting gustatory signals of the brain, first of all, involve the facial nerve. The facial nerve is cranial nerve number 7. And remember, the facial nerve, it innervates the taste buds on the anterior 2/3 of the tongue. Okay, and we're gonna start at an imaginary line in front of the valate papillae. Okay, so the facial nerve is one of those very important cranial nerves. Another important cranial nerve is the glossopharyngeal nerve, which is cranial nerve number 9. This one is going to innervate the posterior 1/3 of the tongue, including the valate papillae. And then lastly, the vagus nerve. Okay, the vagus nerve is going to innervate the taste buds on the epiglottis. All right, any questions with any of that so far?
Can you put that slide back up if you can?
Which one?
The very last one.
Okay, any questions on any of what we covered? So what we have like about 10 minutes, right? All right. Let me see if we can.